Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. That in the last lecture I tried to discuss about the hepatitis B production. I told you that hepatitis B is kind of protein that and it is kind of hepatitis is kind of vaccine that against in the hepatitis B virus. So today I want to discuss another very important biochemical products, and these products is considered as a high value low volume products, and this example is the insulin is greatly used particularly against the diabetes patient. As the time passes on, that we find that lot of diabetes patient, number of diabetes patient has been increases, and then we have, in India we have a vastly populated country, and we have been suffering a lot on the diabetes, and this is somewhat the doctors sometimes say this is hereditarily this is comes in our system, and due to sometimes due to our overtension and in the day-to-day life, and this comes to our system. So this is greatly affect our health system. So due to the invention of this insulin, now it is possible to control the blood level, because which is very much required for this our survival. So I want to discuss how this insulin production takes place. So similar to hepatitis B, insulin also is kind of protein, and this also produced through the recombinant DNA technology. So this only the difference is that it is a two layer. I shall discuss that protein of it is not one chain, it is a double chain, that two layer that is connected with the sulfide bond, and this is how we produce in our through our biochemical processes. So first is that the insulin is considered as a hormone made by the pancreas. Pancreas, if you look at the pancreas, it just it located at the lower part of our stomach, because this is where the pancreas is located, and it allows your body to use the sugar, that is the glucose, from the carbohydrate in the food that you eat for energy and store of glucose for future use. So this has a tremendous effect on the severe different the important effect on our body. Insulin helps to give the blood sugar level for getting from getting too high. What if it is the blood sugar level too high, we call it hyperglycemia, or too low we call hypoglycemia. Hyper and hypo stands for low sugar level, and hyper stands for that high sugar level. Now insulin is prescribed for the treatment of type 1 and type 2 diabetes mellitus. That is the two type of diabetes that we have. Insulin can be given by a syringe. You can see that here how nowadays people they are taking this as their own, and injected pen, they use kind of small pen, and an insulin pump we use that delivered a continuous flow of insulin. So this is how it can be injected in the body. Now the blood insulin level can be measured by the international unit, such as that micro IU. IU stands for the international unit per milliliter, or in molar concentration, such as the pico. This P stands for picomoles per liter, where 1 micromoles IU per ml is equal to 6.945 picomoles per liter. A typical blood level between the mills is 8 to 11 micro IU per ml, which is equal to 57 to 79 picomole per liter. Now, let us talk about the history of the insulin, how it has developed. Now, in the year 1896, Paul, this is Lagerhans, this is a medical student in Berlin, discovered a distinct collections of cells within the pancreas that he isolated, identified that. Then Oskar Minkowski and Joseph von Mering removed the dog's pancreas to study the effect on digestion. Flies are found to be the feeding of the dog's urine, which is shown to contain sugar. Now, in 1901, Eugenie this OP discovered that islet of Langerhans produced the insulin, that the destruction of these cells resulted diabetes. The Benting and Best that improves their technique for the production of insulin, and Eli Lilly, this has become the first who produced the insulin industrially. Now, insulin is sequenced by the British biochemist Frederick Sanger in the year 1955, and is the first protein to be fully sequenced. And insulin became the first human protein to be chemically synthesized in 1963. So, this is the insulin is secreted in our body, in our pancreas is secreted, and if it does not secreted, when you suppose we take some sugar type of product, and if sugar we take, and if insulin secretion is not there, the degradation of the sugar will be affected. So, that is why that insulin dose is very much important. Insulin that concentration in the bloodstream plays very important role. And it can be produced by two ways. You can see that by using E. coli. I told you most of the recombinant protein is usually produced with the help of E. coli, because if you look at different, and this Eli Lilly and company, they use the E. coli. This is the E. coli cell, this is the rod shaped organism that we have, and Nova Nordics, this is another company who use the yeast cells for the industrial production of this insulin. Now, insulin production process using E. coli, how it is taking place, that this is the human cells, and we identify the gene which is responsible for the insulin production, and this is the bacterial cells, am I right? And we take out the plasmid, and this plasmid, this is the gene that we have, we insert into the plasmid, and then we insert into the bacteria E. coli, then grow in culture. This is in the plate we grow, then we grow in the fermentation media. Then after growing, we can extract the insulin from this fermentation broth. This is how the industry that insulin production take place. Now, I was talking about that nature of the protein, that as I told you, as compared to hepatitis B, it is little bit different. Here you look, here if you find that human insulin is made of 51 amino acids arranged in 2 polypeptide chain. I told you there is a 2 polypeptide, this is chain A and this is chain B, and they are connected with the help of sulfide bond. This is these 2 chain, they are connected with the sulfide bond. So, chain 1 having 21 amino acid and chain B having 30 amino acids. So, this is how this is the chain 1 and this is the chain B. the two polypeptide chains are interconnected by disulfide, two disulfide bridge, what we call SS linkage. That this is how this two polypeptide chains, they are linked with each other. This is how it is the formation is there. Now, how this insulin produced in the large scale? The main challenge for the production of insulin using rDNA technology was getting this insulin assembled in a mature form. In 1983, Eli Lilly, an American company, first prepared insulin, I already mentioned before, and two DNA sequence, because I told you two polypeptide chain. The two polypeptide chain, we require two DNA sequence corresponding to A and B chain were introduced in a plasmids of Escherichia coli to produce the insulin chain. This is how, because since we have two polypeptide chains, so at of different length, we use the two DNA sequence, and the separately produced chains are extracted and combined by creating disulfide bond to form the human insulin. They are producing separately and then they combine together to get the human insulin. Now, step 1, because there are several steps involved in the insulin production process. Step 1 is to start the production. Two genes for producing A and B chains are needed, because I have already mentioned the two chains is there. So, two genes are required. One gene produce chain 1 and another gene produce chain 2 B. The step 2, these two DNA molecules are then inserted into the plasmid, because these DNA molecules which code for two different protein, they are inserted in the molecule, and these plasmids are further inserted in the non-harmful type of the bacteria E. coli next to LAC-Z gene. This is the LAC-Z gene is very important. This is kind of this is the arrangement that we have inside the system, and the plasmid also contains the sequence to start the protein formations. So, this is how this insulin production in that take place, and steps for the production in the insulin is that, as I mentioned, that we have two type of that DNA we shall have to find out. So, then we take out this and then we combine, this is A and B, we mix together that and we have the sulfide bond, and we make the A and B is connected with the sulfide bond, then we get the active insulin. So, this is the whole transformation is take place in the E. coli cell. E. coli is considered as the host cell. In this E. coli, this whole transformation will be taking place. Now, the step that we involve, this is by transfection. The plasmids are inserted in the E. coli cell with the help of ligase, and ligase is used for binding purpose, you know that. And then the bacteria contains the plasmid which can synthesize insulin, then undergo a fermentation process. The fermentation is carried out at optimum temperature under suitable conditions. Care is taken to avoid any kind of contamination with the foreign bacteria. So, that is exactly similar to other processes, but only the difference is that there should not be any kind of contamination that should be taking place, so that the growth of your desired organism should not be affected. And in the next step, the DNA extraction is done from the cells by the addition of lysozyme. Lysozyme is a kind of enzyme that break the cell wall, so that whatever the soluble ingredients present inside the cell, they comes out, and so lysozyme digests the outer layer of the cell wall, and a detergent mixture further separate the DNA from the cell membrane. So, we use some kind of detergent just to separate from the cell membrane, it comes to the liquid forms. Let us see here that the human pancreas cells, this is the human pancreas cells, and human insulin producing gene, we take it out, and this is the bacterial plasmid that we have the DNA card with the restriction enzyme, we card the plasmid, and so that we can insert this gene, where this is the insertion of the gene that take place, and then it is overexpressed in the bacterial cells. And when you overexpress, and that way it produces in the fermentation tank, this is the human insulin that is produced, and then this after purification, extraction, purification of the human insulin, we market it in the product as a human insulin. So, this is how it is done. Let me repeat the process again, that this is the human pancreas where the insulin gene is located. So, we shall have to take out the gene, you can see this is the gene, and this is the bacterial plasmid, we have to take out the plasmid, and this plasmid you can count with the help of restriction enzyme, and after that you insert the gene in this particular plasmid, and then you overexpress in the protein, in the overexpress in E. coli, and this E. coli you can, after overexpression, you can have the competent cells, and this competent cell you grow in the fermentation media, so that you can get more cells. You can see they use several cells which has this particular gene, and then it then you cut it, and you can with the help of lysozyme, and then purification you take place, you will get the human insulin. And this is the this is kind of production process that we have, insulin production in Kalundborg in Denmark. This is Novo Nordisk's, the world's largest insulin production facility that we have. You can see you have several fermentation processes are there, and you have both fermentation and downstream processing involved for the production of insulin. Then the bacterium, that DNA is then treated with cyanogens bromide, a reagent that splits the protein chain at the methionine side, and this separates the insulin chain from the rest of the DNA molecules. So, this is very important, that this treatment with the cyanogen bromide, the two chain then mix together and join the disulfide bond through the reduction oxidation reoxidation process, and DNA mixture is further purified through the several chromatography or separation techniques such as ion exchange column, reverse phase high performance liquid chromatography, and a gel filtration chromatography techniques. The different techniques we use for the purification of that particular protein, and the insulin batch is tested to ensure that none of the bacterial E. coli protein are mixed with the insulin, because that this test is to be performed. So, this is very important. This is the kind of downstream processing that we have as far as insulin is concerned. Now, if you look the downstream processing, that steps that is involved in the downstream processing is like this. When you get the recombinant cells, so you prepare the inoculum, what you call seeding. Seeding is the purpose of the preparation of the inoculum, and then we put in the fermentation media. In the fermentation media, we allow this organism to grow, so that it can reproduce this particular organism, and we will get the overexpression of the our desired gene, and then extraction and recovery to take place. Then microfiltration, this is another step that we have to separate the particles on the basis of the size of the particle, and then that optional that pre-purification by chromatography and TFF, Then enzymatic and chemical modification that is take place, and then LPLC or RP reverse phase, that HPLC, that also taking place for the purification of that insulin. And then finally, we do the crystallization and formulation. So, that had, so what I want to stress here, that lot of steps are involved for the purification of that particular insulin, because before it goes to the market, because insulin also directly we injected to the body. So, naturally it is free for any kind of contaminants. So, we take the 100% precautionary measure that the product free from any kind of contaminants. Now, quality control, as I told you, that plays a very important role as per any chemical and biochemical products are concerned. The structure and purity of the insulin batches are tested. So, we mark every batches, that batch number and other things. So, we always test on the basis of the batches, and high performance liquid chromatography is used to determine if there is any impurities in insulin. So, we know that high performance in liquid chromatography is very sensitive instrument. If little contamination is there, it can be easily detected. Now, we know that two type of chromatography technique largely used. One is called gas chromatography, another is the liquid chromatography. Now, in the gas chromatography, we consider the gas as mobile phase as a carrier, and in the liquid chromatography, we consider the liquid as a carrier. So, when we inject the sample, your liquid that acts as a element, it take the material through this column and the separation take place. And this is used just a very sensitive instrument. If any kind of trace contaminants present in your production material, then it can be easily detected. Other separation technique such as X-ray crystallography, gel filtration and amino acid sequencing are also performed. So, these are the different analytical tools that is available just to find out the characteristics of the molecules and try to find out that get the more purified products. And testing of the vial packaging is to ensure the its